From the process of transcription, you will have a strand of mRNA produced. This mRNA contains genetic information from the coding strand. The mRNA will move to the ribosome, the site of protein synthesis, where it will be translated into a polypeptide with the correct amino acid sequence. Now here is how translation happens. This is your mRNA with its base sequence. It will attach itself into the ribosome. Each time, there will be 6 bases that's being read. In your cytoplasm, you have another type of RNA called the tRNA. On one end of the tRNA, you have 3 bases. These 3 bases forms the anticodon. On the other end of the tRNA, you have an amino acid. The type of amino acid depends on the anticodon that's on the tRNA. In your cytoplasm, you have a lot of such combination. Each tRNA carrying a specific anticodon and a specific amino acid. In the process of translation, the tRNA will attach onto the mRNA based on the anticodons they have. The anticodons will attach onto the codons according to complementary base pairing. The importance of this is that you will bring 2 amino acids that's supposed to join together. When they are brought together, they can form a bond more easily. The bond between 2 amino acids is called a peptide bond. The idea is to have a peptide bond formed between 2 amino acids in the right sequence. Now the ribosome will then move on forward to the next 3 bases, or the next codon. After this happens, the original tRNA will leave to bring more amino acids. You will then have a new tRNA coming into the ribosome, forming a bond with the codon of the mRNA, and the amino acids that it brings will be able to bond with the original polypeptide that it's forming. When the whole process is finished for the whole mRNA, you will have a polypeptide with all the amino acids in the correct sequence, or rather in the sequence as determined by the mRNA.